Hello, I'm Sutar, Assistant Professor, YCS Institute, Satara. I'm going to be covering MSc Part 2, Applied Microbiology Syllabus, Pharmaceutical Microbiology. From that, I'll be focusing on Drug Discovery and Development Unit 2. Now, as a microbiology student, I'm sure you must be confused about why we need to study drug discovery and development rather than studying the pharmaceutical or microbiological aspect of the pharmaceutical industry. To make sure you'll understand it quite clearly after this slide, that first, we are in the pandemic situation. All of us are in the pandemic situation. We are listening and we are reading on social media and on newspapers that lots of vaccines and drugs are getting discovered and getting approved. So last month we have discovered this vaccine. They have launched this clinical trial Covaxin. It's the native Indian drug. Then we have received these multiple drug manufacturers have launched these drugs with various pricings. Last month, WHO stopped the hydroxychloroquine clinical trial. Now, hydroxychloroquine created a lot of buzz around COVID-19 treatment spectrum, but WHO stopped it abruptly. So, why did it stop? Then, we all know that Serum Institute is a major pharmaceutical industry in India which has participated with the clinical trials and manufacturing proceedings with Oxford University, and they have started in July itself producing the vaccines. And yesterday, there was a newspaper article regarding COVID-19 vaccine getting clinical trials approved getting clinical trials in India. So, in our day-to-day lives, we also get impacted with the pharmaceutical industry and drug discovery and development process. As an individual who receives drugs, who needs drugs, we need to understand what are all those proceedings that undergo in the drug discovery and development steps. Now, you have seen all the required criteria for a drug to get discovered and to be launched in the market. So first we have seen this is a start or lead identification step. From the first step to the launch of that drug, it requires at least 12 to 15 years of research and development. So for that aspect also we need to understand how the drug development and discovery proceedings undergo. The points that are to be covered in this lecture are going to introductory. So students from the microbiology background, from the chemistry background, or general public can understand from this lecture that the basic concept that undergoes into the drug development and the general terminology that is to be used in the drug development proceedings. So the first Paul Aldrich and the significant terms that are important for the drug development processes are going to be discussed in these lectures. So the important term, first term is drug. A drug is basically a molecule that attaches itself to the various biomolecules. Those biomolecules must be involved in some kind of a disease development, and if we incorporate that drug into that individual or for that disease causing cell or receptor, that condition has to be positively affected. So that is drug. The second is receptor. We know as a life science student that every cell has a receptor on it. That receptor will affect itself, or that receptor has a major impact on cell signaling processes or gene expression processes. So if that certain receptor is manipulated in a way that the disease can be positively affected, then that receptor can be used as a druggable receptor, so that that drug can change the receptor's signaling pathways and the disease can get cured or may get cured. So third is the ligand. Ligand, as we see here, in biochemistry and pharmacology, a ligand is a substance that forms a complex with any biomolecule. In the life sciences, especially in life sciences, ligand is any molecule. It may be an artificial or a naturally present inside your body. That ligand can form a complex with your biomolecule or a receptor, and then can cause or manipulate the signaling pathways. Second is target discovery. Again, we have learned what a receptor is. So this target discovery is nothing but a receptor. We have to discover a particular receptor necessary for that disease to cause. Here we are emphasizing on the discovery of a receptor. Suppose there is a cancer causing cell. Now I have to discover a drug that I need to attach my drug to that certain receptor. So I have to find out the cell that is the receptor on that cell that is the signaling pathway in turn is causing this apoptosis and uncontrollable cell disease. So receptor, and if I find the binding site of that, I'll study that binding site and then I'll or find that will get attached to that receptor and the signal. So that is a target discovery, but finding a receptor that is involved causing process. Target validation is receptor that you have chosen, you have found that is causing the disorder itself. Sometimes in a lot of time it happens that there are multiple parts in that signal. If a particular receptor is causing the disease, it really mean that the only receptor is involved must be multiple enzymes or multiple. So we have to negate all different factors that are affecting the signaling the sole receptor at disease. Compound is a representative of a compound C and potential. The potential is many selectivity, pharmacokinetics, absence of toxicity, and all these potentials are the number of spectrum of compounds, and that is manipulating a very positive and efficient manner that then hit, which has the desired activity in a compound screen and bound upon testing. Hit is lead compound, so you have to find out again a very potential and very effective, so that you will have the 100 drug candidate for the receptor to match. And then lead discovery, again multiple arena or within you have to find out or you have the drug. Lead of the important aspect of the drug. When or once we find the lead compound, then we have to optimize the lead drug so that it will effective receptor binding site. If the binding specificity is only 80%, then the if drug or after the incorporation of drug, so in order to get the 100% effect of that drug, you have perfectly structured binding, 100% specific binding to the receptor to manipulate that receptor. Then candidate selection, again multiple assays that undergo process, and after each selective multiple drugs get selected, and some of them first at the end of that 2 to 3 drugs that are with 100% specificity that can be used to as a drug candidate. Steps are the steps. First, we have target discovery and validation is finding the receptor that is involved in the disease causing process. Confirming that receptor is actually involved in that factor or other receptors are affecting or affecting that process. Second is assay development. Various types of assays are perfect drug or perfect compound, so that we can check the spectrum candidates against our receptors. The for a drug has to be type of the receptor has to be druggable in order for. Then we get 10. Suppose we get out of those 10, we'll proceed. And then we'll get suppose 3 good further investigations. Then lead up selection of drug compounds. We will optimize the structure or the energy level that drug structure, so that it will get binding to the receptor with 100% specificity. Again, the lead optimization, it leads the structure or functional groups of your drug. Shouldn't change the functional groups or the structure, change its native proper receptor. So you have to increase its binding affinity or reduce change as much to change it. Then preclinical trials, discovery and development. After lead optimization, you will get a good binding properties, less toxicity. They proceed with the clinical trials. Preclinical trials are usually held on cell trials. There are 4 phases of clinical trials held in laboratory. Now the approaches, there are 2 approaches when it comes. The first approach is, and the second is reverse pharmacology. In forward pharmacology, first the compound found first, and then you on various receptors or the cells or the diseases mechanism in which the your drug or your compound that you on that cell. In reverse pharmacology, the target first. That means you will discover the target that is involved in that disease causing process. Then you will find that is attaching or that is binding to your receptor. Is found in the stored databases, compounds, various proteins like protein database or various zinc. You can find various proteins or compounds with for that drug that you can use to isolate from that library the drug that is perfect for your receptor. And then comes the lead optimization, where you optimize the drug that you have found from the data bank. You optimize by changing its functional group with rational drug designing, with the use of structure activity relationship. So this is the difference between the forward pharmacology and the reverse pharmacology. They are exactly opposite of each other. In forward, you find you discover the drug first, and in reverse pharmacology, you find the receptor that is involved in that drug. So this is the important part. The reverse pharmacology approach takes about 2 years to obtain a new drug candidate, while in forward pharmacology you will need to find the drug first, then you will find the receptors or the disease that it can act on. Then once you find the disease spot, you will find the in that disease cure. In reverse, it will take just 2 because the receptor and the mechanism is already known, so you just have to find out the drug receptor. Or classical pharmacology is very time consuming. It requires conventional treating. We will see it in the following slide what it is. In a lot of Ayurvedic treatments, traditional ways, Ayurveda has always used the traditional method to discover a drug that can be used. In reverse or target based drug discovery, the method become popular after the Human Genome Project. Naturally, after the Human Genome Project, it is easy to find out the genes that are involved in the disease causing process. It is easy to find out the signaling pathways that are involved in those processes, and it is very easy to find out the proteins that are involved in those processes. So when we get just the gene, from that we will get all the information from the Human Genome Project, and we will know for sure what receptor is to be targeted by our drug. And again, in this reverse pharmacological method, the hypothesis is made that the modulation of the activity of a specific protein target, that is receptor, will have beneficial therapeutic effects. Here, classical pharmacology, first ligand purification, functional activity means first you will find then the scientist will check drug against various receptors, and then biological studies are made. Then this after investigation starts, where the mechanism that is involved in the study. So naturally occurring medicines in all those flowers and plants. Zinca rosea, Sadafuli we see in our has 2 potent anti-cancer drugs that can be used against leukemia, vinblastine and vincristine. This is Artemisia annua. Artemisinin, it gives the medicine artemisinin, Pacific yew bark, which Taxol, the drug Taxol can be extracted from the Pacific yew bark, which is a potent anti-cancer drug. Digitalis, this again is a commonly found drug. Digitalin, the heart medicine, is found in this flower. Then aspirin, the widely known medicine that is used for headache and heart attack symptoms, is found in the Spirea species of flowers. And then Vinca rosea, Sedafuli, we all know this plant. This is the interesting assignment for all those who are interested. These are all the plants with their scientific names on it. So you can Google it and find out what are the important or widely used drugs found in all these plants. Are natural products. The natural products are used by the plants. Those secondary metabolites they are generally made for the protection for evolutionary production of such secondary metabolites. The prospecting. So what is a bioprospecting? Prospecting is a systematic search for biochemistry in nature, really valuable products for pharmaceutical and other applications. Bioprospecting, in a very crude way, when we go in the jungle, we will look for the medicinal plants. We take those medicinal plants in our homes or in our laboratories. We'll extract the important medicinal medicinal drugs or medicinal compounds from them, from various extraction techniques, and we will check those extracted products on various ailments. So that is conventional method of bioprospecting, where we go into the nature and look for the medicines. The phases of bioprospecting start from sample collection. First we go into the nature and collect the samples of the plants like neem or like sadafuli or dhotra. Then we collect those samples. Then isolation, we isolate the medicinal products from those plants with various techniques of extraction. Then characterization. In characterization, the extracted products are characterized with various techniques like XRD, NMR, homology modeling, and then to the product development and launch to the market. Again, an important term you have to know with respect to the bioprospecting part is the Nagoya Protocol. This protocol was made legally binding to the lot of nations in the world. This protocol makes sure that there is a regulation to the access of genetic information of various nations, and there is a fair and equitable sharing of the utilization of those bioproducts. There was an issue happened regarding curcumin or the halal or the neem tree or the basmati rice. All these three species or all these three plants are native to the India, but the foreign nations took patents on all these species, and then we have to pay the money for their use. So to stop all this biopiracy, Nagoya Protocol was made binding to all the nations so that biopiracy is prevented, and the native species or the native people that have been using that knowledge and that have been using those species for a very long time will gain profit from all those knowledge, rather than the pharmaceutical companies or the laboratories in foreign countries. So this is all in the first lecture. This is a very introductory basic lecture. Again, we will see in future lectures what are the techniques that are involved in the extraction methods or the characterization methods, and how the clinical trials will proceed. So thank you.